Okay, guys. Start asking questions about the midterm review. Specifically, this guy right here. All right. If you have any questions about any of the questions that are on here, you can type them in, and I can see what we wrote, and then I can respond. I can try to address any of the questions. I got a bunch of pictures we can look at that are basically the same ones are on this, but I can explain anything that you might be having trouble with. So go ahead, just in the chat, you can ask whatever questions you want. We'll start with one suggested by somebody whose name I can't pronounce, Spinognagi. So someone's got, some type of avatar going on. So I'm just gonna. All right. Oh, it's Spinogai. Oh, it's Jeff. Oh, okay. Yeah. All right, that makes sense. All right, Jeff. Ask about macromolecules. So let's start with that. Let me show you this. Here we go. Okay. So this is one of the pictures we drew in class together. And feel free to ask questions, guys. I won't be able to see them just until I check them, but at the moment, just if you got questions about anything on the midterm review, I can go over the questions on there. Just feel free to ask them. I'll go back and check the comments. Jeff was asking about, I think he was asking about carbohydrates and monomers. We drew this picture out when we started the year with macromolecules. You can see the different monomers and polymers. Not a whole lot on the midterm about these guys. A little bit, I'm not sure what question. Oh, number 13. Which of the following is a monomer? All right, this is number 13 on the review. So if you have the review, take it out, take a look at it. Number 13 gives you 4 options: triglycerides, DNA, amino acids, and proteins. It's asking which one is a monomer. So you can see on the right here, monomers include like fatty acids, nucleotides, saccharides. Monomers are more like anything that's like bigger, like triglycerides, polypeptides, DNA, and starch. So when it gives you the options, triglycerides, that's not an option because that's a polymer. Triglycerides are polymer. DNA, also a polymer. And again, you can see at the top of this chart here, polymer. So DNA, triglycerides, those are not an option. Then it gives you amino acids and proteins. So proteins, you should know, are pretty big. Your best option is amino acids. These are monomers. All right, amino acids would be monomers. So be sure to look over this picture. We drew it earlier in the year. That should help you. So yeah, you gotta know which ones are monomers and polymers, absolutely. All right, what else we got here? I got a bunch of screaming emojis. Someone Gisella's about to fall asleep. And a bunch of highs. So you guys want to ask questions, like legit questions, or I can go to sleep. Nothing so far. All right, well, DNA stuff. DNA stuff. Hi, Jesus. How's it going? All right, DNA stuff. Just because no one's saying anything else, so let's go over some DNA stuff. The main things you gotta know about DNA are the structure. You gotta know the ATCG stuff. So A pairs with T, and C pairs with G. And it would help, guys, if you do have questions, it would help to identify a specific question that you're asking about on the review. So let's say you want to know about DNA, like number 6 and 7. You look over the test and figure out which questions relate to your topic you're wondering about. So this is number 6 and 7. 6 is asking which of the following is a correct pairing in DNA, and that answer for 6 is D because that's A and T. You can see B says U and G. Just so you guys know, U is only found in RNA. All right, not DNA. So that's impossible. You wouldn't if they're asking. If I'm asking you about DNA, you wouldn't talk about you. Don't even mention you. Number 7, saying during DNA replication, a DNA strand that has the bases CGAT produces a strand with which bases? So just writing where in the room I have here, they give you CG, or I give you CGAT. So what's the proper pairing? It's CGCTA. I give you this, you have to pair it up and figure out that this would give you that. All right, you definitely have to know that. That will definitely be on the midterm, which is tomorrow. Let's see what else you guys got here. Prokaryotics versus eukaryotes. Yeah, we can do that in a second. Anything about, Michael's asking about cellular respiration specifically. All right, so I'll get to that in a second. Light dependent versus light independent reactions. Okay, so let's start with prokaryotes versus eukaryotes, and then I'll go to cellular respiration really quick. Let's see if I have any pictures about prokaryotes versus eukaryotes. So the questions, I forgot which questions address prokaryotes and eukaryotes. You definitely want to know organelles. I know that. You definitely want to know like the structures and functions of all different organelles. This is the only picture I have that's closest. Like know that prokaryotes are like bacteria. Definitely know that. Eukaryotes are like us. Our cells are eukaryotic cells. Main thing that defines a eukaryotic cell is having a nucleus, other organelles. Prokaryotic cells have free-floating DNA. You just want to know the different structures that are found in prokaryotes and eukaryotes. The other questions, let's see. Cellular respiration. You want to know the products and reactants for cellular respiration? Let's see here. Do I have one on that? So glycolysis, know what goes in, know what goes out. I had one with mitochondria. Let me find that find that really quick. 1 second, guys. I'll show you this. I have one more picture I gotta show you that will help if I can find it. Yeah, I don't see it. I'm pretty sure I have one like it. Oh well. You basically got to know the products and reactants of each phase of the cell cycle. Yeah, I could have sworn I put one on here. Anyway, glycolysis, for example, you can see that let's go on there. You can see that produces, we can remember that produces a total of net of 2 ATP and glycolysis. The other ones, you got to know, again, the products, all about the products and reactants, all about what's going in and what's going out. Know that the cell cycle requires oxygen. Know that it requires energy in the form of glucose. And know that what goes out is CO2, lots of energy, and H2O. You want to know that reaction, the products and reactants of the cell cycle. That's, I believe it's one of the questions. I just forgot which one. Let's see what else we got here. Light dependent versus light independent. Let's talk about those really quick. Good place to go with that is chloroplasts. So if you recall, there's a picture of something like this on the test. You got to know. When it comes to light dependent versus light independent reactions, I'm just going to draw this little corner here. You got to remember that picture we drew. You should definitely know that picture. And you had on one side, you had the thylakoids stacked up. On the other side, we had the Calvin cycle. On the left is the light-dependent reactions. They obviously need light. And then on the right, you got the Calvin cycle, which needs carbon dioxide. The Calvin cycle, or the light-independent reactions, so this is the independent side. So be sure to look at this picture. We drew it together earlier this year. Be sure to review this guy. This side makes glucose, and this side makes oxygen. That's basically it. That's basically everything you need to know. Know which side is which, know what goes in, what goes out. That's basically it. Electron transport chain. The main thing you need to know about the electron transport chain is how much ATP is generated. And where it takes place. It takes place in the mitochondria. It makes a total of 32 ATP. Oh, the other thing you want to know is what it requires. It requires oxygen, and it's used to make water. So know that about the electron transport chain. What goes in, what goes out. And that's it. That's mainly it. All right, some other questions here. The functions of different macromolecules. All right, so going back to the macromolecule, Shantunu's got a bunch of good questions here. I appreciate that. Back to the macromolecule picture. The functions of these guys. So monomers are like starch and monosaccharides. They are just sugars used for energy. Carbohydrates. Nucleic acids, that's just your DNA used to store genetic information. Nucleic acids also includes ATP. So not only is it DNA, but it's also ATP. Those are both like nucleic acids, categorized as nucleic acids. Proteins do everything, absolutely everything. And they're enzymes, so they help perform reactions in your body. And then lipids also help store energy in the form of triglycerides, fats. They also help make your membranes, your cell membranes. That's the main function of lipids, is help make your cell membranes, and storage of energy as fat. What else we got here? All right, Ryan's asking about alcoholic fermentation, lactic acid fermentation. Know that only we perform lactic acid fermentation, whereas yeast perform alcoholic. You want to know the products and the reactants of each. Know that lactic acid fermentation, it's basically making like the lactic acid, as a part of our cellular respiration. And alcoholic fermentation is obviously making ethanol or a type of alcohol and CO2. So you want to know like what's going in and what's going out of each one. Okay, autotrophs and heterotrophs are categorizations for organisms. Autotrophs are organisms that make their own energy, like plants, and heterotrophs are like us, things that they consume for energy. So that's a good question from Gisela. Shantunu is also asking, do we need to know the products and reactants for each stage of cellular respiration? That's probably the main thing you have to know about cellular respiration, like what's going in and what's going out. Callie's trying to figure out how to type. Let's see here. Do you have to know the microscopes? No, that will be on the EOC, but it's not on the midterm. So we'll review microscopes again at some point, but it's not going to be on the midterm. Like that's stuff we definitely have to know for EOC purposes, but I didn't put it on there. Michael's asking acids versus bases. Really quick, I'm going to find a picture of acids and bases to help you guys with this. Give me 1 second. Because the question we had on here, and then I'll hit up number 21. Someone mentioned number 21. That's the heterotrophs, autotrophs. So I'll go over it anyway, in a little more detail in a second. So just trying to find a picture of acids and bases. The question asks about hydroxide ions and H ions. So let me show you guys what that's all about. Give me 1 second here, guys. Just trying to get this working. I can use this picture. Okay. So a new picture for acids and bases. I just looked one up really quick. And this is actually a pretty good one for the question that we had on the, again, this is number 9, number 8, number 9. So bases have these this right here is called hydroxide. That's something you want to know just for the rest of your high school life and college life. Hydroxide, whereas these guys up here are hydrogen ions. All right, technically they're both ions, hydrogen ions and hydroxide ions. They're both ions. This guy makes you more basic. If you have a bunch of OH ions floating around in a solution, it's a base. If you have a bunch of H ions floating around in a solution, it's an acid. That's the technical, like, chemical definition for each one. Lots of hydrogen ions means you're acidic. Lots of OH ions means you're basic. The other question is asking about the pH scale. So if you're going, acids are basically 1 to 7, or I guess really 6. Bases are 8 to 12. And then 7 is categorized as neutral. All right, other questions. Just a couple more things here, and then we can, let's see. There you go. Sean Zanino, I hit up that. Yeah, there you go. We're talking about less than 7 for acids. So you guys got that covered. I forgot what bleach is. I forgot if bleach is an acid or base. I don't know. Krebs cycle. I think the only thing you have to know about the Krebs cycle is that it produces carbon dioxide. I think that's it. You want to know that pyruvate goes in the Krebs cycle. So what goes in, what goes out? That's basically it. So know that pyruvate goes in, carbon dioxide comes out. Oh, so the example. Okay, so that's why you mentioned bleach, Catherine. That makes sense. So you don't have to, the examples from pH scale. No, I don't think the examples on there. You just need to know the definitions of acids and bases. So that's a good question. I'm glad you mentioned that. Michael's asking about chromosomes. Yeah, use that number 5. We labeled that in class. Definitely, just know the 2 like structures on a chromosome. That's really it. We'll talk more about chromosomes in January. You know, Ibby, that's a very good question. Peanut butter cups exist. Why not have almond butter cups? I bet they'd be delicious. I'm sure they exist somewhere. You just gotta look for them. So that's a very good question. And if you can't find them, you should invent them, because that's a $1 million idea. The citric acid thing. I forgot what the citric acid. Oh, that's just the same thing as the Krebs cycle. That's another name for the Krebs cycle. That's all. So just to address Callie's question really quick, she was mentioning citric acid. Let me go to the picture of that. I think I have a picture of that on here. The closest thing would probably be, yeah, I guess I don't. I thought I got most of the pictures that were on here, but I guess not. This guy, glycolysis picture. Because at the end, everything, like the pyruvic acid, is used in the citric acid cycle. So glycolysis is the very first step of cellular respiration. All of the products, the pyruvate, then moves on to the Krebs cycle, which is equal to the citric acid cycle. Same thing. I mean, for the citric acid cycle, you want to know what goes in and what goes out. For the Krebs cycle, what goes in is it's pyruvate. Pyruvate's going in. It's right here. And the main thing coming out is CO2, carbon dioxide. That's really it. What else we got here? Cohesion, adhesion, and water. Good question from Charlie. If that's on here, I don't know where it is. I don't think that's on the midterm. That might be on the EOC, but I don't think I put any of those questions on the midterm. Cohesion is like attracting, or like being attracted to like, whereas adhesion is like two different substances having an attraction. It's really, I wouldn't worry about it too much. It's a good question, but you don't have to know it for this. Catalysts versus enzymes. That's a good question. So catalysts and enzymes are basically the same thing. I wonder if I have. Yeah. So we basically drew this picture in class. It's the midterm has a very similar picture to this. You guys can see it. It's right with number 15. Number 15 has a picture just like this. Enzymes are the same thing as a catalyst, just a definition. A catalyst is basically something that speeds up a reaction. That's easy. Enzymes are proteins. Catalysts can be a bunch of different substances, but proteins are a biological catalyst. That's it. Okay. G1, G2. Jeff's asking about G1, the G2 thing, and then Shantu's asking about diffusion, cell diffusion. All right, let's hit up those two things, and I think we'll be pretty good here. So first the cell cycle stuff, and then I'll go back and talk about diffusion. This picture right here. So these are all the phases. We labeled them in class. G1 phase is like copying organelles is the main thing. S phase is copy DNA. G2 phase is prepare. And they're just preparing for mitosis, process of mitosis. Obviously, you gotta know all the phases of mitosis. There you go. And G1S, G2, that's interphase. That like G1S and G2 make up interphase, whereas mitosis is its own separate phase with PMAT and then cytokinesis. Shantanu asked about the diffusion facilitated and facilitated diffusion and just passive diffusion. Really, the only thing you want to know about that is mainly the active transport requires energy. All the other ones do not. And also know about the types of solutions, which is kind of related, the types of solutions and what happens to a cell in each solution. Because like basically what's happening in a normal cell is you have diffusion occurring at a constant rate going in and out, whereas in other types, like a hypertonic cell or a cell in a hypertonic solution, you have osmosis occurring really quickly into the cell, making it explode. In a hypertonic solution, you have osmosis occurring really quickly out of the cell, making it shrivel up. So not a whole lot with active transport and diffusion, just knowing the basic definitions of them. Let's see here. So I just mentioned that from the Infinite Ranger. I'm sure that's a made-up name too. Okay, do you have to know smooth ER and rough ER? Yes, good question, Ryan. Smooth ER makes lipids, rough ER makes proteins. Remember, rough ER is covered in ribosomes. Smooth ER looks literally smooth. It does not have a covering of ribosomes. Michael's asking about how the mitochondria has H2O coming out. Can I explain that? Sure. That's a very good question. If I can find. You're just going to have to bear with me for a second, Michael, because I got to find a good picture of that to explain it. So give me 1 second. And I just found a picture right here. This will have to do. So the main thing when it comes to mitochondria is knowing how cellular respiration occurs in mitochondria and what process of cellular respiration occurs when. So I just found this picture online. Just a quick search. And it's basically the one we drew in class. So it's cut off, but whatever, it'll work. So you got glucose is going in. Michael was asking specifically about the water. What's going on with this water over here on the right? Why is water involved? Well, water is made in the electron transport chain. So when you're like the last part of cellular respiration is called the electron transport chain. That makes a total of 32 ATP. And that also makes water. It's already up here. It also makes water. So you gotta remember there's 3 phases of the cellular respiration. The first one's glycolysis. And then you got the Krebs cycle occurring, also known as the citric acid cycle. Krebs cycle makes carbon dioxide, and then you got the electron transport chain. So glycolysis is 1, Krebs is 2, electron transport is 3. Yeah, that's cellular respiration in a nutshell. Just trying to keep it really simple and quick here. Okay, last couple questions, and we can close it out here. What happens? So Jeff is asking what happens in prophase, metaphase, all those different phases. You definitely gotta know that. You want to be able to look at a picture, like the picture on number 10 of the handout of the review, and the picture is a lot like this one here. You need to be able to look at the basic, what's going on basically in each picture and be able to identify. So you need to look at like this one of prophase and see how like the nucleus is starting to break down. See how you can see chromosomes? That's a big sign that prophase is occurring, that you have these big chromosomes form. Another sign that spindles are forming. And we talked about that in class. You need to be able to like see when these guys are lined up, when the chromosomes are lined up, that's obviously metaphase. All right, so once you see these chromosomes lined up, you need to know that's metaphase. Anaphase is when they split apart. Telophase is when you have almost 2 new cells, and then you have 2 actual new cells in cytokinesis. So you gotta know each of these phases. You'll see a couple questions on that for sure. So that's a good question, Jeff. Okay. Ibby, what's your question? I'll wait here a second. Endergonic versus, so Shanae's asking about endergonic versus exergonic. I'll get to that in a second. Lactic versus alcoholic. And we mentioned that before. You just need to know that we perform lactic acid fermentation. Yeast perform alcoholic. Know what goes in, what goes out. We've drawn that out. Know that alcoholic makes ethanol, and that it also makes CO2. All right, guys. I don't carry my phone around with me like you guys do. Like I don't have no idea. If you text it to me, I have no idea what you sent me. Like no idea. So hold on, Ibby. Let me go back. Let me see what your question is. I mean, I already addressed the almond butter cups suggestion. So, I mean, I don't know what other questions you could have. Okay, the graphs. Last thing is someone mentioned endergonic. Catherine mentioned endergonic and exergonic. So let me hit that up in the graphs, and then I think we'll close out with that. So last thing. So here's one of the graphs. Something like this is in the midterm practice. So when you look at this graph right here, you need to know that this is an exergonic reaction. This is a reaction that releases energy. You have energy released. Energy released. We call this exergonic. So this is the main one you're going to see on the test. I might show you endergonic reactions, which is where you have it flipped around like this. Something like that. So you want to be familiar with that one too. Also notice this right here, how you have a difference. That's because of activation. That's called the activation energy, the energy required to start the reaction. and also know the reactants are on the left, products are on the right. That's basically it. That's really it. All right, I think we're pretty good here. Anaerobic means just does not require energy, or excuse me, oxygen. That's all. Shanti, what's your last question? You haven't asked it. Structure of DNA nucleic acid pairing. I hit that up earlier in the video, so if you missed that, you can go back and watch it. And I think we're good here, guys. I think that's it. I think you guys are ready for this midterm tomorrow. Let's see here. There you go. Okay, so tomorrow I'll see you guys in the morning, first thing. I don't see any other dire questions. If you know anyone who missed this review, it's going to be up. Like they can catch it. I posted it on Classroom too. So if someone missed it, not a big deal. It's up there. You guys can watch it whenever. All right. Get some sleep tonight. Get up and early. I'll see you in the morning for that test.